Thanks everyone for being here today. I'll try and keep us on time for coffee. So I'm going to be talking today about ecological interactions and mostly focusing on microbial ecosystems. But some of these ideas that I'm going to be talking about may also be applicable to macro-scale ecosystems such as plant communities. And if you're interested in this, please do ask me at the end of the talk. So as Alejandro very nicely introduced in his talk, microbes don't live in isolation. They're always in communities in lots of different contexts. And if we're thinking about microbial communities from a functional point of view, the composition of these communities is really important. So function follows on from composition, because different microbes have different metabolic capabilities, and they are able to contribute different roles to these communities. So we really want to be able to manipulate community structure in a lot of different contexts. So maybe we want to introduce, for example, a nitrogen fixer to the soil, or maybe we want to remove a pathogen from the human gut. So in order to be able to do this, we need to think about the ecology of these systems, because these are really ecological questions that we're asking. How is it that we can ensure that a species is able to invade an existing community, or how can we ensure that another species is driven to extinction? Now, the way that people have kind of tried to approach this is using classical ecological theories, where we consider the interactions between different species. So when I say an ecological interaction, what I mean is a growth rate impact. We have the effect of one population on the growth rate of another. And these can either be between different populations, or they can be within populations. So the effect of one species on its own growth. You can then take these pairwise interactions and assemble them into large models, where we then do a bottom-up prediction of community properties such as resistance to invasion and whether or not species go to extinction in the long term. And the kind of framework that people have used to do this in the past are these Lotka-Volterra models, where we have these interaction matrices which represent these networks of interactions between different species. Unfortunately, in microbial systems, this has been running into difficulties in recent years. So this is not exactly a test of this Lotka-Volterra framework that I was talking about, but it's sort of related. It shows how pairwise relationships don't necessarily assemble into community properties. So in this study, they looked at they took these stable assembled communities and then broke them apart into these individual pairwise components. And what they found was that the pairwise elements were not predictive of the community level properties. So in general, you found that you had competitive exclusion of one of these species in these pairwise assemblages. So this kind of bottom-up assembly maybe is not quite as simple as we might hope. So why might this be happening? Well, one idea might be that we have context dependencies of these interactions. So maybe where we're in this community context, we have a very different interaction network to what we have where we're in these pairwise contexts. So if you're interested in ecological context dependencies, Switzerland is a great place to be because there's a lot of research in this area currently going on here. So in our own group, there was this study from a few years ago where we were looking at these batch culture systems where we put resources and cells into a flask and basically shake it and then measure the interactions at the end of the experiment. And what they found here was that there was this environmental context dependency whereby, depending on the composition of the environment, you see more or less negative or positive interactions. And in general, you find that there are more positive interactions, for example, when you have toxins in the environment compared to when there aren't. Then there's this work from Martin Ackermann's group where they find that there are these temporal dependencies in these batch culture systems. So you find that there are changing interactions over time. And then in the group of Alex Passat, they were studying these microfluidic chips where we have unidirectional flow through a flow cell, and we see that there's spatial structure emerging within this system. So in this, you can see that there's this orange species that's growing downstream of the green species here, and that's indicative that the green species is having an increasingly strong positive impact on the growth of the orange species as we move along the length of the device. So that's indicative of a spatial context dependency. So what this project is about is trying to understand where these context dependencies are coming from and how we can predict them and maybe exploit them for practical purposes. So I'm going to use a theoretical framework to approach this, which is slightly different from the Lotka-Volterra model that I was looking at before. Instead of considering these direct interactions, we're going to have interactions mediated via environmental change. So microbes are always interacting with each other by manipulating their environmental composition. So we, in general, have a separation of our world into these species and our environmental compounds. Species can impact these compounds through these impact functions. So these can be either positive impacts, so production of compounds, or negative, which is depletion or degradation of compounds. And then we also have these sensitivity functions in blue, which describe how these compounds impact the growth rate of a target species alpha. And again, these can be either positive or negative. And we can now assemble these sensitivity and impact functions in all the different possible combinations of the signs, and we get these 4 different elementary kind of interaction mechanisms in microbial ecosystems. So we have these positive mechanisms, enrichment and detoxification, so this is kind of cross-feeding, if you're familiar with that term, and then we have these negative interaction mechanisms of depletion or nutrient competition and pollution or intoxication, so this is kind of antibiotic production, for example. Now we can take these elementary components and build up to more complex metabolic interactions by generating these composite interactions and depending on the exact elementary units that we've used to build up these composite interactions, we can have different net interactions. So these 2 cases are fairly simple. We just have positive interaction mechanisms or negative interactions present in the system. But this is the more interesting case where we have a mixed case. So there's both a positive interaction mechanism of toxin degradation down here, but also a negative interaction mechanism of resource competition up here. And so depending on the relative concentration of these 2 compounds, we might expect to see different net interactions arising in this system. So this is where environmental dependencies seem to come from. So I'm going to now talk about an example about how this actually works. This is kind of the simplest possible example of one of these mixed interaction systems, where we have a single species A that's interacting with itself via nutrient depletion and then toxin degradation down here. And I'm going to represent the concentrations of these different compounds with these environment spaces where we have nutrient concentrations and toxin concentrations on the 2 axes. And then I'm indicating here this quantity which is called the instantaneous interaction, which is simply the composition of these impact and sensitivity functions that we're using to describe this ecosystem. And you can see that we have positive and negative regions here, and these represent the varying balance of these 2 mechanisms in different environments. So we see up here that we have domination of this toxin degradation positive process, and then down here we have domination of this nutrient depletion. So this is capturing the idea that there are different dominances of the different interaction mechanisms in different environments. Now, in these batch culture systems that I talked about before, we generally start off with a particular environmental composition, and then we move through this environment space, and we have some sort of trajectory through time. So we start off here, but then we move to this kind of endpoint where we've depleted the resources in this system. And so we might predict that we have a change in the interaction sign from positive to negative over time. So here we have an experimental model of this theoretical model that I was showing you in the last slide. We have this single species, Commomonas testosteroni, which is competing with itself through proline and degrading ampicillin in the environment. So this is kind of the toxin degradation component. And we're going to ask, is this something that we actually see? Do we see a switch from positive to negative interaction values? So we can put this into different environments containing different concentrations of ampicillin and proline, which is going to allow us to tune the strength of these 2 mechanisms. And then we can measure the interactions over time. So to start with, looking at this case where we have no ampicillin in the environment, so this toxin degradation pathway can't really be expressed, we see, as predicted, that there's no positive interaction phase because there's no ampicillin in the environment. But when we do have ampicillin in the environment, we see that there is a shift in the interaction from positive to negative as we predicted through the theoretical model. And the strength of that positive interaction phase becomes stronger as the ampicillin concentration increases. So comparing this now between all the different experimental conditions in the theory, looking at the peak positive and the final negative interaction value, we see that there's this nice match between the 2 sets of results. So this is capturing temporal and environmental dependencies, but what about spatial dependencies? So to look again at these microfluidic chips that I was talking about before, where we have this unidirectional flow through a system, what's happening here is that we have fresh media coming in, and then the composition of this media is changing as we move along the length of the device. So we have a very similar idea to what we had in the batch culture, in that the environment is changing as we move along the length of the system. I'm going to explain this example using this example ecosystem. So we have 2 species now, a degrader and a crossfeeder, and they're interacting via these 2 metabolites, a polymer and a cross-fed metabolite. So it's a slightly more complex ecosystem than we were looking at before, but it's still amply described with these environment organism models. Now we can provide a similar analysis as we had before, so we can derive these instantaneous interactions. But now instead of just having one environment space to describe the interaction, we have 4. So this kind of corresponds to, if you're familiar with these Lockwood-Volterra models, the interaction matrix. But whereas normally we would have just single values for these, here the interaction matrices have been expanded into the whole environment space. So this is capturing these environmental dependencies of the interactions in different environments. And now again we can have this, we have these diagonal elements which are the kind of intraspecific self-interactions, and these off-diagonal elements which represent the interactions between species. So now again we can have this batch culture model that we have here. So this is with the dashed line, and you see that again we have this environmental trajectory through the space. So we're again expecting to see time dependencies of the interactions in that system. But we can also have this microfluidic model, which is very similar, but instead of looking at time, now we're looking at space. So we're moving from the inlet over here to the outlet over here. And again, you can see that we have this changing context, which is going to lead to changing interaction values. To cut a long story short, when we now compare the temporal dependencies and the spatial structure that arises from the spatial dependencies in this model to the experimental data, we see again a nice match between these two sets of data. So to conclude this part of the talk, we have this framework, which I'm going to call the closed environment organism system, because the system is closed with respect to environmental inputs. And in this system, we find that we have these elementary interaction components, which can be assembled into more complex models. The relative strength of different elementary components determines the net interaction value that you actually measure. So we'll call that the environmental dependencies. And then these time dependencies and spatial dependencies are essentially just environmental dependencies in disguise. They result from changing environments as species modify their environment autonomously. And this entire thing that I've been talking about for the last 7 minutes is amply described by this equation here, which we can derive from those elementary mathematical components that I was talking about before. So this is a description of the dynamics of these systems under closed conditions. So that word closed is a slightly tricky one. If you're trying to think of a slightly critical question for this discussion session, a very obvious one would be, who cares about batch culture? Because, I mean, batch culture has nothing to do with real ecology. Maybe there are some very odd sort of marginal cases where batch culture is of interest, but in general, this is a very unrepresentative experimental system for looking at microbial ecosystems. We put everything at the beginning, and then we just let it evolve until it exhausts all the resources. Really what we want to describe is an open system, such as a chemostat, where we have continual inflows of resources and effluxes of material. So what this system has is a dynamical equilibrium, which can be described with those Lockwood-Volterra models that we described before. Unfortunately, although this is more realistic in a lot of cases, so if we're thinking about the human gut, for example, you can imagine that we're taking in food, so that's kind of what I mean by these environmental inputs. There are some downsides to using these chemostat systems. So they're tricky to work with, they're low throughput, it takes a long time for these systems to equilibrate, and you kind of have to run one every week, whereas you can do hundreds of these batch cultures every day. Secondly, the environmental composition of these systems is unknown, because even if you know what you're putting in, microbes are very messy eaters. They tend to secrete a lot of metabolites. And even with modern high-throughput metabolomic techniques, it's very hard to assess actually what the environmental composition is. So if you're trying to take into account these environmental context dependencies, it's actually very hard to use these chemostat systems. By contrast, in these batch culture systems, you do at least know what you're putting in at the beginning. So you have some information about what the environment consists of. So this is sometimes what I call environmental dark matter. It's stuff that we don't necessarily know, but probably is playing some ecological role. So basically what the approach that we're going to take in the future, and we're not doing this just experimentally yet, but some theoretical work allows us to approach this, is that we want to use these advantages of the batch culture system to more amply explore the chemostat system. And so we have these 3 sets of equations which describe these 3 different types of ecological system. I don't want to dwell on this too much, but basically I just want to point out that there is a precise formal relationship between these notions of interactions between these different systems. And what that allows us to do is it allows us to take a chemostat system. So this is a simulated chemostat. We have these equilibrium environment, so I'm going to call that R star. We can then put that into this batch culture system and then evaluate these interactions at that equilibrium environmental state. And we don't need to know what R star is, so we kind of overcome that problem of the environmental dark matter. We can then plug this into the Lotka-Volterra system and then make a prediction of the community composition at steady state. And this is just a plot showing this in theory. So with 500 randomized communities, we do see that there is some initial signature of correlation between these two different quantities. So we can predict, in principle, chemostat steady state composition using only batch culture data, which is much easier to acquire. And so longer term, what we're hoping to do here is that we want to use this batch culture as a kind of intermediate step where we explore all the context dependencies of these interactions and then use this information in order to tailor the environment to manipulate community composition, as I was saying at the beginning. So this is kind of closing this loop at the beginning where we were wanting to manipulate community structure. In principle, we should be able to do this more effectively by having this intermediate step and then changing the input to these kind of chemostat systems in order to manipulate the composition and thereby the function. So to summarize this talk, I've been talking about these EO systems, environment organism systems, where we can split between organisms and their environment, and then we can put these into many different types of experimental system. And by unraveling the links between these different types of experimental system, we can more deeply understand community composition and dynamics. So in part one, I talked about the link between these two systems, and in the second part, I talked about the link between batch culture and chemostats. So with that, I'd like to thank the research group. If you'd like to look at the first part of this talk, we have a preprint here. Many thanks to the HFSP, of course, for funding this project. And I'd also like to thank Stefano, who is in the Stocker group in ETH, who has been helping with some microfluidics, which I'd be happy to talk about at the end, but I didn't have time to talk about today. So thank you very much.
Thank you very much. A few questions and then much needed coffee.
This one.
Oh, thank you. This is super interesting. I was wondering, in your framework so far, are you mostly allowing for kind of constant interactions, so consumption of a nutrient at a constant rate? Or could this also be, to some extent, environment dependent? I'm thinking of things like pH or stuff like that.
Yeah. So these impact functions and sensitivity functions are extremely flexible. So you can put basically any environmental factor into them, and it will just get. I mean, you have to construct them carefully, of course, but you know you can build them up from those components in kind of any way that you like. So yes, you could manipulate those factors based on pH. Also, if the pH is being changed by the species, then that can also be reflected in these models. Yeah, so they're very flexible in that respect.
Okay, thank you very much. And let's thank also